Welcome to today's webinar, which is titled Gene Therapy for Neuromuscular Disorders. On behalf of Muscular Dystrophy Canada, I'm really pleased that we have today's webinar as we have received a number of questions on gene therapies over the past year and especially in the past few weeks and months. Really pleased that we have Dr. Miriam Oskley, Dr. Jim Dowling, and Dr. Hugh McMillan. They are leaders in the field of neuromuscular research and neuromuscular disorders. They practice clinically in pediatric settings and have been involved in gene therapy clinical trials and other disease-modifying therapy trials as well. So without further ado, I'll pass it over to you, Dr. Dowling, to get started. I know you'll be presenting some information. We have received your question. We'll dedicate some time towards the end to answer those questions. Over to you.
Great. Thank you, Mira. So we're going to do three relatively quick talks to hopefully set the stage for answering all of the great questions that have come in. And my task is just to quickly introduce gene therapy so that we're all talking about the same thing and to highlight a few of the principles that I think answer some of the questions that have come in. So just to note, I'm a clinician scientist at the Hospital for Sick Children in Toronto. And these are some of my disclosures. I think the most important one is that there is a pre-existing educational initiative called the Gene Therapy Network, and many of the slides I have been taken from that. This network is now no longer active, so but they have some nice educational material which I'll be using. So just wanted to, at a high level, talk about the idea of genetic therapies, which are obviously quite important and impactful related to neuromuscular disorders. And when we think about genetic therapies, we're not only talking about, you think back to those of you from high school biology, the idea that you have DNA and DNA in every one of your cells, and DNA makes RNA, which is then processed to eventually be made into protein. And the mutations that cause neuromuscular disease can occur at the DNA level, but then impact the production and processing of RNA and or the expression or function of protein. And therefore, you can imagine that a therapy targeted really at multiple parts of this pathway could be potentially effective for treating neuromuscular disorders. But today we're going to be focusing specifically on a DNA-based therapy. And when we talk about gene therapy, we really are talking about DNA-based therapies, and essentially ones where the idea is that you're restoring a DNA that is otherwise not functioning properly. And in general, and this is based really on the FDA definition of gene therapy, there's sort of four major areas that are covered. The most common in the way that we're really thinking about gene therapy is the idea of gene transfer. And that's where one has a missing or non-functional gene that is replaced by that same, a normal version of that same gene. There can be gene augmentation, which is where the idea is that therapy targets increasing the expression of an existing gene in the genome. Conversely, gene inhibition would be the idea of using a therapy that targets the knockdown or reduction of a gene that's producing a toxic RNA or toxic protein. And then finally, gene editing. And I think people are probably familiar with the idea of CRISPR-Cas9 gene editing, and not something we're going to be discussing today, but is a therapeutic strategy and one that's being considered for Duchenne muscular dystrophy. But again, we're going to be focusing on gene transfer today as the main topic, because the microdystrophin gene therapy is a form of gene transfer. So in order to understand gene therapy, it's really important to understand what goes into it. And these are the essential components really of any gene replacement therapy. The most important thing is what's called the transgene. That's the actual gene that's now being introduced into a patient, in a patient's cells, that contains the gene that hopefully is providing the therapeutic benefit. In the case of microdystrophin, the transgene is a miniaturized version of dystrophin. For other gene therapies, it may be the full gene. And I'll talk a bit about why it's a microdystrophin versus the full dystrophin. There's what's called a promoter, and that's a sequence that enables the transgene to be expressed at a high level and specifically in tissues of interest. And then this whole thing needs to be packaged into something that can successfully deliver it, in the case of Duchenne, to skeletal muscle and to heart. And in most cases, it's a viral vector that's responsible for this delivery. But just to comment on the fact that there are multiple different kinds of delivery mechanisms, Right now, the standard in the field for gene therapy is using a viral vector, but there's emerging data about non-viral delivery systems, which may ultimately be impactful for numerous neuromuscular diseases, including Duchenne. But right now, these non-viral delivery systems are very difficult to get to muscle and have a lot of limitations that still need to be overcome before they can be utilized. And that's why you'll see that most gene therapy clinical trials and gene therapy products are using these viral vectors, which enable very efficient delivery that produces good gene expression that can last a long time. Importantly, we'll talk about some of the cons, including some safety concerns, a key limitation in the size, and these can be very difficult to manufacture, and that impacts a bit the availability and also the price of gene therapy. There are many different kinds of viruses that can be used for gene therapy. Right now, the standard in the field is to use what's called adeno-associated virus, or AAV, and that's for a couple reasons. The main ones are the safety components related to AAV versus other viruses. And the big thing that's been shown is that AAV, when introduced into a human cell, does not integrate into the patient or person's existing DNA. And that's important because for some of the other viruses, it's been shown that they can integrate into the DNA and then potentially either produce harm because of that or promote over the long term the development of cancer. And so a non-integrative delivery system is really important. Another is that it has long duration, which is important, particularly for a skeletal muscle disorder, and also has a relatively favorable side effect profile. So these are all features that make AAV attractive. A key limitation to understand is the size restriction. So 4.6 refers to the number of thousands of base pairs that can be put inside the AAV. That's 4,600. To give you a context, that's about less than half the size of the full-length dystrophin gene, and about half the genes in the genome are larger than 4.6 kb. And so that's an important restriction when thinking about AAV-based gene therapy, and the rationale for why for DMD, the strategy has been to use a small version of it because it has to fit inside this AAV packaging. Most, as I mentioned, clinical trials are using viral vectors. I'm just going to skip past this. It's important to note that there are multiple different kinds of AAVs, and these are naturally occurring viruses, and so they've been co-opted because of their low, because the fact they don't make people sick, but they also can infect human cells. Different AAVs will have different predilections for different tissues in the body, and therefore different uses. For skeletal muscle, AAV 8 and 9 have been the most commonly used, and that's because they target the muscle and the heart well. And the version of the gene therapy that Sarepta has developed that we'll be talking about more was based on a derivative of AAV 9. One other important thing to note is the way that AAV is made. So I mentioned that you have your gene with your promoter and it's all packaged inside of a virus, but then it has to be produced. And that's done using often a mammalian or insect cell line. And what happens is one puts these components inside the cell, and then the cell basically spits out the AAV virons, which then are used as the therapeutic product. And it's important to know this for a couple of reasons. One is that this is a limitation in terms of the manufacturing capacity, this requirement to do this. And number two is that depending on the cell that is used, different modifications will happen with the AAV. So this can be a potentially underappreciated source either of the trophism of AAV for different tissues, or else the potential for side effects related to AAV. In general, AAV can be delivered in two different strategies, and gene therapy in general can. One is ex vivo, where you take cells out of the body, give the therapeutic product, and then put them back in. This is something that's being done quite successfully for blood and immune disorders, but probably at this point less applicable to skeletal muscle diseases. And then there's the in vivo delivery, where the gene therapy product is given so that it can get through a large portion of the body. Was originally some consideration of the idea of using intramuscular, where basically a direct injection into the muscle, which can give a high amount of AAV. The challenge there is obviously for DMD, you'd have to inject many muscles. And so that proved not to be feasible. So instead, we have used an intravenous route to deliver the AAV. I'm going to skip this, which is basically, I think the important thing to note is that the AAV enters the cells through receptors that are on the cell. It then goes into something called the endosome where it's broken down. Then the DNA that's inside of it will go into the nucleus where it then is able to make the RNA and protein that is important for treating the disease. As probably people are aware, there's a lot of excitement about gene therapy. And there's dozens of clinical trials ongoing and hundreds of gene therapy programs in preclinical development. And neurogenetic diseases are really among the most common that are being targeted with gene therapy. This is just a few examples of some neurogenetic diseases. And neuromuscular diseases have actually been pretty at the forefront of gene therapy, which is great for our field, but has also meant that we've as a field, been a group that has really experienced a lot of the learnings about gene therapy, both the good and the bad. One of the questions I know came up with the idea about improvement and sustainable improvement. So this is just to mention that there have now been several different clinical trials for gene therapy products for neuromuscular disease. People have probably heard about the gene therapy product for SMA. There's also been an ongoing trial for a rare muscle disease called X-linked myotubular myopathy. And obviously today we'll talk about the DMD clinical trial. But the question of duration of gene therapy is really obviously very important. If a child is being dosed at age 5, and the hope is that the child will live for many decades, the gene therapy has to last for that long. And at this point, we don't really know how long in humans gene therapy will be sustained in skeletal muscle. If one infers from studies done in large mammals like dog models, it seems like it's at least several years that the gene therapy will persist. And while the expression may wane over time, it does seem to be a quite durable therapy. And I think it's just going to be a matter of learning over time to know how long that will be and what to do after gene therapy may wear off. And I think there is some expectation that eventually the expression will wane to the point that it's no longer meaningfully producing the gene therapy product. So just to conclude with some considerations that have emerged from gene therapy trials, I'm not going to really talk about CRISPR, but there are some interesting ethical and accessibility and inclusivity considerations related to genetic therapies. Who should get them, what diseases should they be made for, and how do we ensure equitable access for these expensive therapies to all relevant patients. Safety is a definite consideration, and I think something that has been sometimes minimized in the advertising or discussions related to AAV. It is in fact a virus, and it's given in quite high doses. And so it's somewhat not surprising that patients can have clinical symptomatology related to that. I've just listed a few of the things that have occurred in patients. Fortunately, these side effects have largely been not significant or severe, but it is not always the case. And there have been individuals who've had quite severe adverse effects from the AAV product, including some deaths really across all of the different AAV-related clinical programs. I already mentioned about durability, and we can certainly get into that further. There are important questions about how one can combine gene therapy with other therapies that are coming, and whether that makes sense to do and what that might look like. I think probably everyone is aware that these are not insignificant therapies from a cost perspective. And this is just the cost of the SMA gene therapy, and we anticipate that the DMD gene therapy will also be several $1 million per dose. And then I was just going to conclude by mentioning, because I know this came up in a couple of the questions, the adverse events are clearly dose related. The higher the dose that one gets, the more likely one will have adverse events, and the more likely those adverse events will be severe. And this factors into the reason why there are age and size restrictions to who can get gene therapy. And that's because of this concern about a dose threshold for safety. Should mention that the adverse events are likely related to immune response primarily, to either the AAV itself or to the gene that's being delivered. And so the idea of immune modulation is certainly going to be something that's considered over time, both to hopefully minimize adverse events, but also when we think about repeat dosing. I think I will stop there. And let me stop sharing. Thank everyone for attention. I'm going to hand it over to Hugh.
Terrific. Thanks, Jim. I appreciate that. I'll just do my best to promptly share my screen. And I hope everyone can see it. If not, please let me know.
We see it in presenter mode, though.
Terrific. Let me see. Presenter mode.
Oh, where you had just
Oh,
I just stopped sharing and I'm just going to try again. Is that any better?
that's great.
Okay, terrific. Thanks for your patience. So I'm going to spend the next 5 to 10 minutes talking about the very practical matter of access to these new therapies. And I'm going to touch on some of the key scientific phases and key elements in the regulatory process in Canada, and briefly comment on a slight difference in the US. I have just a few disclosures to note, advisory board memberships, and then the work that I've done as the principal investigator of clinical trials. So we'll touch on 6 key areas with regards to access to new and emerging therapies. I'll start on the left side of the screen, the orange box, and talk about early research. And that's a very key area that we look to our colleagues in basic science to carry out. And these sometimes can have very little to do with the actual disease we're studying in the end. They sometimes can ask much broader questions, such as, can we manufacture viral vectors? How are we able to make them? What are different types of viral vectors? And then, as Dr. Dowling was commenting on, can we fit the gene inside the viral vector that's going to use? So there's been a tremendous amount of research that's been done over the years to try to help us prepare for the clinical trials that we're seeing now. There's the preclinical research that's being done as well after that early research. And those tend to be a bit more disease specific. They'll often begin with cells or cell lines and then move to mice and then eventually to higher mammals. They provide very important information to us about safety matters, side effects that can be seen with these emerging treatments. They give us information about the dose that would be predicted to be used in humans, and how mammals and their bodies break these drugs down. So when a parent or a doctor is looking at these protocols and looking at these clinical trials, it's very important that we don't gloss over these preclinical research studies, and that we ask very important questions such as, what were the side effects in the mice or in the rhesus monkeys or whatever the animal model that was used to try and understand how these therapies may work in humans. Then moving into clinical trials, the key features that make these clinical trials first is they involve human beings. So the work that I've talked about before, the preclinical work, those were in animals and animal models and cell lines. Clinical trials is when we move that research into the clinic and the emerging therapies are used sometimes for the first time on humans that have underlying diseases. Clinical trials also compare the effect of an intervention, such as a drug or a gene therapy or a device, between groups. Often there will be a placebo group that's part of the clinical trial protocol, a group that will not receive therapy, but one that looks and mimics it in every way. Sometimes, and this was the case for some of the spinal muscular atrophy trials, is when we have a very good understanding of natural history, sometimes it can compare it to natural history groups, and it can be an open label study where everyone gets the drug. The other feature that's important about clinical trials is they're, by definition, prospective, meaning a drug or a therapy is given, and then the person is studied as we move forward in time. Phase 1, phase 2, and phase 3 trials, which I'll describe in the next few slides, all require government approval for them to be run. In Canada, Health Canada must receive a clinical trial application and have no objection to the trial being run. In the United States, it has to be the FDA, and in other jurisdictions, there's other governmental bodies that need to approve it and make sure that it's acceptable for people to receive these in clinical trials. There also needs to be a trained and qualified investigator, and she or he need to have staff that can carry out everything that's required in the clinical trial. And then there also needs to be research ethics board approval so that an entity in the hospital is also in agreement with the trial being run. Now, to talk about phase 1 trials, these are early safety phase trials. These are the first in-human studies that are being carried out. For many drugs, these used to be carried out on healthy volunteers. But in the case of gene therapy, because of ethical issues, and it wouldn't be ethical to give a healthy volunteer a gene therapy, the phase 1 trials will involve children or adults who actually have the disease that the therapy is aimed at trying to improve. The purpose of phase 1 trials is the very first assessment of safety in humans. Another goal that it has is to determine the safe dose range. And it looks to see how does the body react to and break down the drug or therapy? How is it metabolized? Safety is really the primary outcome for these phase 1 trials. Phase 2 trials are called the early efficacy phase trials. This is when the drug or the therapy is given to a larger number of people with the disease. Traditionally, these phase 2 trials would involve several hundred people. Now, with rare diseases like SMA or DMD, phase 2 trials involve a much smaller number of people, but they're much larger than the phase 1 studies that I spoke of earlier. And the goal of the phase 2 trials is the very first test of potential effectiveness or efficacy of the therapy. Ongoing safety and side effects will, of course, still be monitored, but this is where there's the first attempt to have a larger group and look to see if there's any evidence that the drug is making a difference in terms of outcome for the people who receive it. Phase 3 trials are confirming the efficacy of the drug. And traditionally, they would involve a very large number of people with disease or a condition, usually in the thousands. And again, with rare conditions, it's not possible to have numbers like that, but they do need to be large enough for us to truly be able to see that the efficacy, if it's shown to be there, is convincing and truly is able to benefit patients. Again, there will be an ongoing assessment of safety and side effects as part of the larger phase 3 trials, but efficacy is the main focus of that. And phase 4 trials, I'm not going to talk about too much because they occur after a drug has been approved by Health Canada or another regulatory agency. That's the post-marketing surveillance phase. And the purpose of that is to gather more information about the drug, the long-term benefits, and potential longer-term risks that the patients who've received the treatment might experience. Sometimes it's run as a trial, sometimes it's run as post-marketing surveillance through a database or a registry, for example. Now, one of the things we start to see with some of these rare diseases is there's combining trial phases. And people may have heard of this, and Dr. Asqui may talk about this with some of the DMD trials, where it's not clearly cut into 1 and then 2 and then 3, but rather there will be an attempt due to the rare nature of these diseases to combine them. So it's not unusual to hear about a phase 1/2A trial where safety will be the focus, but there also will be some an eye to very potential early efficacy of the drug, or a phase 2/3 trial that will try to look at the potential efficacy of the drug in a smaller number of patients. After these trials have been run, if the data looks promising, if there's no very severe or very worrisome side effects that are emerging, if the data appears to be beneficial, then the company or the sponsor must go forward and try to get regulatory approval. And in Canada, the company would need to apply to Health Canada for a new drug submission. And they apply to Health Canada's Health Products and Food branch. There's two options with that. Health Canada does have an accelerated review when there's an unmet medical need, and they also have a standard review. The accelerated review may take 7 months as the target. The standard review can take 1 to 2 years. Health Canada requires solid data from phase 3 clinical trials as part of their review process. Now that differs a little bit from what we see now in the United States, and it's one of many reasons why companies will file typically for approval in the United States first. The US Food and Drug Administration, or the FDA, now have something known as an accelerated approval program. And what that program allows for is it allows for the earlier approval of drugs that are used to treat a serious medical condition. So SMA, DMD would certainly fall within that category. And it also has to fill an unmet medical need based upon a surrogate endpoint. And by surrogate endpoint, they will allow for and accept a laboratory measurement such as high CK in the case of Duchenne muscular dystrophy, or for other diseases, a radiographic image like an MRI that's believed to predict clinical benefit. So for example, if there was a drug or a gene therapy that allowed for CK to drop considerably, then that surrogate endpoint could be taken in an earlier trial, an earlier phase of clinical trials, and allow for a company to apply to the FDA for an accelerated approval of their drug. Now, the FDA would still require those drug companies to carry out the phase 3 studies, and they would look at that data to make sure that the effect is still efficacious and it's still safe. If it's not, or if the companies don't provide that phase 3 data eventually, then the drug could be removed from the US market. So just to show an example, if there's a phase 1, phase 2A study, in the United States, if that data looked favorable, if there was a surrogate endpoint, even though they may not have yet demonstrated significant clinical benefit, the US would review that submission from the company. Whereas other jurisdictions, such as the European Medicines Agency or Health Canada, they would require more information, more data before there is a likelihood of them being willing to accept that application and perform that review. Once Health Canada has approved the therapy, it's not yet available for people to be able to universally access. After Health Canada has approved, and in some cases, during the early phases of the Health Canada review, there's two separate agencies called CADTH and INESS that need to also then review the data. CADTH stands for Canada's Drug and Health Technology Agency, and Quebec has an equivalent called INESS that reviews that data on an approved drug and makes a recommendation to all of the provincial governments. In the case of INESS, they're making a recommendation specifically to the government of Quebec as to whether or not they should provide reimbursement for this treatment. And if the answer to that is yes, for which subset of patients should potentially be reimbursed from the provincial payers to receive that treatment. So Health Canada, for example, in the case of SMA, gave a very broad approval and said that omnisemnogene abeparvovec, or gene replacement therapy, can be used for spinal muscular atrophy. But then CADTH in INESS put much stricter criteria on who could be reimbursed for using that. Once CADTH and INESS recommendations are made, many private insurance companies will follow along with those recommendations as well. Now, it's important to remember that that takes additional time. So even after Health Canada has made their additional review, which can take 7 months if it's accelerated, 1 to 2 years for a standard review, there's additional time for CADTH. And in the case of omnisemnogene abeparvovec, the gene replacement therapy for SMA, CADTH published their recommendations 9 months after Health Canada had approved omnisemnogene abeparvovec to be used for SMA in Canada. There's two special considerations to keep in mind. So the first dot that I have here is prior to Health Canada approval, when a drug has been approved in another jurisdiction. Now, Health Canada does have a special access program, and that special access program can allow a treating physician, it must be completed by a treating physician, to file an individual application for a drug or treatment that has been approved in another country to potentially be used in Canada. Now, the major problem with that is, number one, is that even if Health Canada says, yes, you would be allowed to bring that drug in for it to be used in Canada, it is not possible to receive any reimbursement whatsoever from either public or private insurers. It's not yet considered a drug in Canada. It does not have a drug identification number, so it is absolutely not reimbursable in any way. The other piece that's very important is that the application must be made by a treating physician. So it has to be a therapy that the treating physician believes is safe, that she or he believes that there's enough data that it's efficacious, that they would feel comfortable placing that application and feeling that it's in their patient's best interest to receive that therapy. The other indication, or the other consideration, is after Health Canada has approved, but before CADTH or INESS has made their recommendations. In some instances, private insurance companies may cover the cost of the drug after Health Canada has approved it, but before CADTH and INESS have made recommendations to the provinces and the territories. But I must emphasize that provincial payers will not pay for a drug until after CADTH and INESS have made their recommendations, and the province has made a decision themselves as to who they feel should be reimbursed within their jurisdiction. So these are considerations that may not be a path forward, particularly for very expensive drugs, such as gene therapies. There's two documents that I can point you to, a therapeutics product regulatory process in Canada summary, as well as a second useful resource that can be available in terms of the review process in Canada. So I'll stop there, and I'm going to stop sharing and hand things over to Dr. Asqui. Here we go. I think I did it. I'm not very technologically savvy, but I think I did it. I'll turn it over to someone who's much more technologically savvy.
Oh gosh. You guys all seamlessly shared your slides. Pressure is on for no malfunction. How does this look?
Looks great. Thank you.
Awesome. All right. So with Dr. Dowling providing a beautiful framework of what is gene therapy, and Dr. McMillan walking us through the different study phases and how drugs come to market and can become accessible to individuals, I was tasked to briefly digest the recently published study on a gene therapy product for Duchenne muscular dystrophy. I'm an investigator for several trials and work as a methodologist for the AAN on their guidelines subcommittee, but I don't work as a consultant. So I obtained most of my information from two sources, the first being the peer-reviewed publication that came out this summer that I'll talk about, but also the slide deck presentation that Sarepta put together and presented to the FDA. So, some of the slides are outdated, but hopefully useful visually and better than what I could create for you guys. So we're going to talk about the SRP 9001 clinical development, focusing, I don't know if you can see my mouse here, on study 102. When the FDA application was placed, really all three of these studies were put on the table, this one being kind of closed for enrollment, and the results had not been available yet, but it was going to be upcoming. And since then, now we have Embark, and this is outdated. There's now Envision as well in other studies. The first one really just was 4 patients, very, very initial proof of concept. So study 102 is the placebo-controlled trial that was recently published that I'll talk to you guys a little bit about, so that you can put it into a framework and see what you all individually can make of it. So this is the reference. I think it's open access. So what is it? Essentially, it's a gene therapy product of a truncated dystrophin that is introduced and in development for Duchenne muscular dystrophy. And it is a phase 2 study, but there were 2 parts. So the first part really is the placebo-controlled part, where the children were randomized to either get the infusion of the gene therapy or infusion of a placebo. And the outcomes are measured. There's 2 primaries that we'll talk about over that span. And then in a blinded way, then they're crossed over in the second part, where those who had not gotten the gene therapy now get it. There were 2 sites in the US for this study. And they did stratify, meaning, they included kids 4, 5, 6, and 7, but they made sure they had equal kind of numbers of both groups of patients. And so the first part is 48 weeks. They do a biopsy after about 3 months, and that's one of the primary outcomes. And then they do the functional motor assessment at 48 weeks to see the change from the beginning. And that's the other primary outcome that was measured. And they had about 40 boys in this study. And I think it's important here, let me even go back here because this one's a little easier. So, if you're trying to look at a study to say, is this drug going to cause this outcome being, improvement in motor function, essentially, in the boys with Duchenne muscular dystrophy, the best way to do that is to, to take a child, give them the treatment, measure things after 1 year, and then go back in time, take the same child, give him, the placebo, and then measure the same treatments after a year. And the reason for that is you want to make sure that all the other factors that could have an impact on the outcome you're measuring are controlled for, right? It's the same person. We know that across boys with Duchenne muscular dystrophy, no 2 boys are the same, right? And that's the truth for so many other neuromuscular disorders as well, right? So the reason why it's important in these studies that allocation, so how we decide who gets the treatment and who gets the comparison group, if that process is random, meaning no one can control the order in which, who's going to get treated or not, is that it helps kind of control for things that you can't even think of measuring, right? Because there's things we can measure, things we can't measure. So really important that randomization occurs. And then you also want to then look, depending on how many, if you only have 4 people in your study and you randomly assign them, it's highly likely that there'll be some uneven distribution of some important things between the 2 groups, right? If you have more people, chances are it'll be a little bit more balanced. So I'll get to this point in a little bit. So what was this particular study looking at? Well, the population, it was, boys with Duchenne, 4 to 8. The treatment was this intravenous single dose of the gene therapy. Comparison group was actually a placebo infusion. And the outcome, there were 2 primary outcomes, so 2 main outcomes they were looking at. The first one was measured at 12 weeks after that infusion to compare kind of the expression of that truncated dystrophin protein. And the second one was to change from baseline in the North Star ambulatory assessment, and that's the functional motor assessment. And that was at 48 weeks. The boys were all on a regular dose of steroids for at least 3 months. And then that dose the day prior to infusion was increased to at least 1 per kilo. And I mentioned this because I think it's interesting to note that in their calculation, the way they decided to have 40 people in this study was based on the fact that they said, well, if we want to have our primary outcome be the North Star assessment, we want to be able to measure a 5-point difference between those who get the drug and those who get the placebo. And so, you can do a little bit of math and figure out how many people are needed in the study. The North Star, for just those of you who are more or less familiar with it, essentially looks at different, motor tasks, rates them on a scale of 0, 1, and 2. You can do with difficulty or you can't do it. There's a loss of that function. And we know that there's a lot of heterogeneity, especially in younger boys, in terms of increase and decrease and changes in that function. I'll skip over this. Looking at the distribution in the younger kids and the older kids, one of the points that did come up was that the randomization was not as successful in the sense that the older boys, those who initially got gene therapy versus those who initially got placebo, they weren't very similar. They were a little bit different in that those who got the placebo were higher, had a higher level of functioning. In terms of the outcomes, the primary, remember the first one, they wanted to see on muscle biopsy, is there expression of this truncated protein? And sure enough, in that first part where you can compare it to the placebo group, there was a significant difference in the expression on those muscle biopsies of about 23% in those treated. And when you followed them and you repeated the biopsy a year later in the second part, well, they still had that like 19% expression. So didn't further increase from there, but it was sustained. And for the North Star, that was their other primary outcome, it didn't reach significance. So putting all the boys, the 4, 5, 6, and 7-year-olds, because that was their intent to compare with the placebo arm, there was no difference at 48 weeks. But then they separated into age groups, and this wasn't a primary outcome, but just to explore, saying that, well, maybe if those baseline differences were different in the 6 to 7-year-olds, maybe that could kind of explain some differences. And they found that in the younger kids, there was a 2.5-point difference, and that was kind of mathematically significant, versus in the older boys, there was not an important difference between the two groups. I'm looking at the time. So these are some of the secondary changes of looking then in the second part that maybe we can skip over. And adverse events that were reported as being related to the treatment. The most common ones were, decreased appetite, nausea, vomiting, and most of the adverse events that were reported in the study were in that first part, in the first 3 months of treatment. There were some events of rhabdomyolysis that resolved, some increases in serum transaminases, that also then resolved, no deaths. And then in the second part, there were no treatment-related serious adverse events that were reported. So I think I'm going to stop sharing there of my very quick overview of Study 102, because I really want to give time to some of the questions that had come up from all of you. So we do have some designated time for that.
Wonderful. Thank you so much, Dr. Osqui, Dr. McMillan, and Dr. Dowling. Very clear presentations. I'm already seeing a lot of positive comments in the chat in terms of really helping to better understand gene therapies and how they work and clinical trials. What is nice that I can see already is we received over 60 questions on gene therapy, and through your presentations, many of those questions have been answered. But I'd like to go through some questions, and I'll start with you, Dr. Dowling, if that's okay. You had a slide whereby you spoke about when gene therapy could potentially fade, wane over time. And there was a question that there have been some information shared on the DMD gene therapy that Dr. Osqui just spoke about, that it potentially could fade after about 5 years. And this parent is asking, if that were to be the case, then what happens? And is there any experience in other disorders of what happens when a gene therapy fades?
Yeah, it's a great question, and also, one that I think is on everyone's mind in terms of how that may impact patients. I think at this point, any estimate on how long it lasts is not going to be accurate because it's probably inferred from non-human studies. And we just don't know. There haven't been individuals dosed for long enough with biopsies to look at expression over that length of time to know. If you infer from some of the dog studies, I think the gene therapy has persisted for more than 5 years, upwards of 8-9 years, definitely with a drop, at least a 50% or more drop in expression. But it's hard to know because so much is actually expressed from these constructs, whether that actually is consequential. But if we assume that that would be the case, then I think it's really hard to know what that might mean, whether if you have sustained the gene therapy for a number of years that you would then, and you can imagine a scenario where then you'd start to have the disease progress again when it's no longer there to provide, the protection that it does, or perhaps the muscle reaches a state where it's less exposed to damage, and so the process wouldn't be the same. I think it's a really, really difficult question to answer. And one other thing I would mention, and this factors in also to the questions about the age at which we talked about, there's probably a ceiling right now in terms of the maximum dose you could receive that's safe, and that may limit the age at which you could get it from an older age. But there's also a concern about dosing too young in terms of, think about how much muscle you have when you're 6 months old versus when you're 6 years old. And the muscle compartment's still actively growing. There's the potential for diluting out the gene therapy impact. If your muscle is doubly as large, you have half as much gene therapy there in a way. That's a concern certainly that's been addressed or in other diseases where the tissue is more proliferative, like in the liver. Like there's concerns about doing gene therapy too young for liver disease because the feeling is that it would be diluted. So a lot of things that still need to be worked out. And I think it's important to appreciate the window that's been looked at, as Dr. Skew just mentioned, is a very narrow one. And so outside of that window, it's really hard to know what this is going to look like, whether it'll work, whether it'll be safe. And that's why the studies really still need to be done.
Dr. McMillan, in the recently FDA approved gene therapy, it was mentioned for age 4 to 5. We did see that study information. In the case of SMA, a lot of families talked about that they were told earlier is better. And so this parent is asking that my child is 3 years old with Duchenne muscular dystrophy. Would the same principle apply? Earlier is better, as in younger than 4 years old, as indicated in that FDA approval, which we know that does not have a Health Canada approval. But if we go off that same criteria, would earlier be better?
It's another very good question, and the challenge with that is we don't have an answer for it. It's a bit like some of the other therapies that are moving forward in clinical trials, such as vamorolone, for example, where there was an initial study that was carried out on a certain age, and then there's follow-up studies that are bookending or looking at younger or older boys to see if it's efficacious. Earlier does tend to be better for therapies, particularly gene therapies, because younger children, particularly very young children, potentially may have less immune response. But the challenge with this is that we need to also give it at the right time. So we need to give it at the time that's going to have the most beneficial effect for, in this case, the boy, because we don't necessarily want to give it if it's too early, and we may not see the maximal benefits. So we don't know when is the optimal time right now. And I do feel that even after these clinical trials are done, we will need to look at other ages, at other side, to see if there's a benefit and what that benefit is, and whether it's better than the age bracket that's been studied in the current trial by Sarepta.
Thank you. Dr. Osqui, this is a question from a parent who says that my son, did not qualify for the Pfizer gene therapy trial because he developed antibodies for AAV9. So in this case, would he not be eligible for other gene therapies, particularly the one by Sarepta that you had shared the study on?
So not necessarily. I think the antibody titers would need to be re-verified. And there are different titers that are checked across depending on what the gene therapy is packaged in. So not necessarily would be my answer.
Dr. McMillan, if it's okay, I'll ask you this question, and it's from a parent who has a child with SMA who has received gene therapy, Zolgensma, and they are now asking whether there are any studies or evidence to show that they should be followed up with nusinersen, Spinraza, or risdiplam as part of maintenance.
In Canada, the neuromuscular specialists are following really the evidence, and right now the evidence shows that being treated with therapy, either Zolgensma or an ongoing nusinersen or risdiplam treatment, are effective. There's no evidence, though, that adding a second treatment on offers any additional benefit. So that therein lies the challenge at the present time. There are some clinical trials for spinal muscular atrophy that will recruit children that have had Zolgensma and potentially offer them an intrathecal treatment with an antisense oligonucleotide, a drug that's very similar to nusinersen or Spinraza, but those are just beginning in clinical trial right now. So at the present time, the recommendation, if in Canada, if a child's had Zolgensma, is that they not receive nusinersen or risdiplam unless there's signs of wearing off effect, which to my knowledge, I haven't heard of. So we would not recommend an add-on therapy in that case. It's different from the US, but Canada follows most of the other jurisdictions in other countries around the world in terms of that recommendation.
Very clear. Thank you. Dr. Dowling, this is a question for you. A parent was saying, actually, as they entered today's conversation and webinar, they thought gene therapy and stem cell therapy were the same. Can you actually tell us how those are different, stem cell replacement therapy and gene replacement therapy?
Yeah, so they're a little bit apples and oranges in that, that gene therapy is a vector or delivery-based strategy where something is packaged within a delivery and given to cells, and then cell therapies where the actual cells are given, and the idea being in this case that the muscle has stem cells within them that carry the capacity to grow and expand the muscle. And also to repair the muscle in response to injury, there's long been the thought that healthy stem cells could repopulate muscle that's affected with muscular dystrophy. The challenge with cell therapy is really getting the cells to the muscle, and there have been several clinical trials trying to look at delivering satellite cells, and they don't get taken up by the muscle very well. And so haven't proven to be effective. I think there's still a lot of hope related to a satellite or a stem cell based therapy, but still some technological hurdles that need to be overcome. You could imagine an ideal scenario where you would use, so for DMD, the idea behind stem cell therapy would be to take someone else's stem cells and or muscle satellite cells and deliver them, because that would then have dystrophin. But you could imagine, like the dream scenario being that you fix the dystrophin in a patient's satellite cells and then redeliver them. There is work underway to try to do that, either using gene replacement, but or else potentially CRISPR to correct the mutation and then redeliver the satellite cells. But again, that's sort of combining gene therapy with cell therapy, but they're two different things. And right now, cell therapy for muscle disease isn't really been successful.
Very good. Dr. McMillan, this is a question based on your presentation on the special access program. When you mentioned that this parent has a child that's 7 years old, and they're saying that if they were to go through the special access program to apply for a drug that has a criteria or a restriction in age for another country, is that what the special access program is for? That it takes, for example, the FDA approval for 4 to 5, or could a 7-year-old, a 6-year-old, a 10-year-old with Duchenne also be eligible to receive a drug through the SAP?
Health Canada would review the application if it was submitted. I don't know what their decision would be because it would be up to the medical reviewers of Health Canada to decide. But I would say it's very unlikely that they would approve a drug to be brought into Canada to be used that's being used off-label for another indication for the same indication, but for a different age group in another country. But it ultimately would be their decision. The other piece to that, though, is that one would have to find a physician in Canada to make that application, and I think physicians would be very, very reluctant to dose a child with a medication in Canada and put that child to potential risks associated with that when it's off-label, unapproved in Canada first, and then off-label in another jurisdiction.
Very good. Dr. Oskey, to your knowledge, is there any evidence on whether families affected with boys affected by DMD should probably do exon skipping therapy first and then look at gene therapy, or the other way around? Is there any recommendations or worldwide or evidence that really points to how to make that decision?
I would say that there is no evidence to support doing one before the other or vice versa. And seeing the time, I think that's the extent of that response.
You know, Howard, I'd love if we can end this conversation. Actually, a lot of the questions are from families really wondering how to get gene therapy or how to make that decision. So I'll turn it to you just in one sentence. If you were to offer a tip or advice to a family member that's trying to make a decision for their child with DMD or spinal muscular atrophy. There's families here with Nemolan myopathy who are asking. So what would you say? How do you make that decision of when to get gene therapy, what gene therapy? There's more than one gene therapy for DMD under development. So Dr. McMillan, how do you kick us off with your tip?
Yeah, it's not an easy answer that could be answered in one sentence. I think there needs to be a discussion with that child, that family's treating neuromuscular specialist, because gene therapy for one disease is not the same as gene therapy for the other disease. It's not as simple as what are the complications to the viral vector. Some diseases have cardiac involvement that could increase risks. Some have liver involvement that could increase risks. So the individual disease really needs to be considered in the context of what the gene therapy is and what the potential benefits are, balanced with the potential risks.
Great. And I do see that Dr. Oskey and Dr. Dowling are nodding in agreement. So with that, I will go ahead and end today's webinar. Really thank you for your clear presentations. I know these are difficult questions as we're really getting more and more information day by day, and there's a lot of emerging evidence, but we welcome patients and families and community members to continue to send these questions to research@muscle.ca, and we'll of course always connect to experts like those that are on the line today to get you the right evidence-informed answer. So thank you all so much for joining today's webinar, and we wish you a good afternoon, good evening. Thanks everyone.
Thanks for having us, Samara.